Hello and welcome to Biology Interest. Today we're going to be looking at the kidneys and the nephron in terms of the filtration of water and other minerals in A-level biology for AQA. So as you can see, I've got my diagram of a nephron, of which there's about 1 million of in each kidney. These are very important in the reabsorption of minerals and water back into the blood. So it's got 4 main areas we're going to look at. And that's the glomerulus, which we'll look at in a minute first. The proximal convoluted tubule, which selectively reabsorbs stuff like glucose, etc. Loop of Henle, where a sodium ion concentration gradient is created to reabsorb water. And the distal convoluted tubule, which just kind of reabsorbs everything else to maintain a balance. As well as the collecting duct, which is the path to the ureter, and also where more water is reabsorbed essentially. So first we're going to look at the first step of filtration in the kidneys, and that's the process of ultrafiltration in the glomerulus and Bowman's capsule. And so what essentially happens here is that the blood arrives from the renal artery in a thicker arteriole essentially, and then it splits into a whole bunch of very thin capillaries, and then it exits via another arteriole which is actually thinner than the entrance arteriole. And what essentially this causes is a higher pressure to build up in these capillaries here, because the wider arteriole entering is wider than the exit one, so it kind of causes a block up of blood essentially, which causes the pressure to be higher and push out harder. Now, in a similar way to the way tissue fluid works, which you've probably looked at before, this capillaries causes a higher blood pressure, and that high pressure, or hydrostatic pressure as it's called, causes particles or molecules that are small enough to be able to pass through the capillary membrane, to actually pass through that membrane and into the Bowman's capsule. And so that's essentially what happens and is called ultrafiltration in the kidney. And what passes through is water and ions that are small enough to go through this membrane, and stuff like proteins, which are too big, remain in the blood, causing it to have a low water potential, which is important for the reabsorption of water later. And so now we've got all our glomerulus filtrate. It enters the Bowman's capsule and passes down the proximal convoluted tubule. And what essentially happens here is substances are selectively reabsorbed depending on whether they're needed and whether they are wanted. So for example, urea, which is toxic, is not really reabsorbed anywhere along here, but especially in the proximal convoluted tubule, which contains loads of microvilli to allow us large surface area for selective reabsorption. Stuff like glucose, amino acids, and other important mineral ions are essentially actively transported and moved by facilitated diffusion by proteins back into the blood, so they're not lost through excretion, which is very important to maintain the levels of those in the blood via homeostasis to make sure we can still function. And so this causes essentially like stuff in the urine to be water, dissolved salts, urea, and other substances that we don't really want or have too much of. Now, one thing to note is urine doesn't usually contain proteins and other big cells, and also glucose. And so if there's a sign of those in the blood, if you get a question like that, it's likely to talk about diabetes or kidney failure, and so be aware of that. You don't really get proteins or glucose further in the blood than the proximal convoluted tubule, because especially glucose is selectively reabsorbed. So now we've looked at this, we're going to be looking at the loop of Henle, and we've got another diagram for that. And so this is an important part of how water is reabsorbed in the blood. And so what essentially happens here is that we have these two tubules, the descending one and the ascending one. And what happens here is sodium ion gradient is created to cause water to move out or be reabsorbed essentially. And so I'm going to draw on my diagram to show you what happens. So this is the ascending limb, it's going up, and this is the descending limb. So what happens on the first step is on the ascending limb towards the distal convoluted tubule, we have sodium ions, pumped out by active transport using ADP into the medulla, which is the area of the kidney that the loop of Henle is in. And so they are basically pumped out by active transport. And now the water, and so essentially this lowers the water potential in the surrounding area around the loop of Henle, lowering the water potential. Now, the ascending tubule is actually impermeable to water, so the water can't diffuse out by osmosis to balance the concentration which is being changed by these sodium ions diffusing out. And so what actually happens is, because the loop of Henle, the two tubules are so close to each other, the increase in sodium ions causes water to diffuse out by osmosis out of the descending tubule. So essentially water diffuses out here into the medulla and then gets reabsorbed back into the capillaries by osmosis because they have a low water potential as well, due to the high concentration of proteins that were left inside when ultrafiltration occurred. And so that keeps happening there. And then when you get to the bottom, because the water keeps being removed as you go down the descending tube, the concentration of ions, sodium ions, inside gets really, really high. In fact, it gets so high when it reaches the bottom of the loop of Henle that the sodium ion concentration is higher than the concentration of sodium ions in the surrounding area. And so these sodium ions then diffuse out of the bottom of the loop of Henle, further increasing the levels of sodium ions in the medulla, meaning more water diffuses out by osmosis on the descending tubule and is reabsorbed back into the blood. So anything left over, even water, continues back up this ascending tubule, and because it's impermeable to water, nothing else leaves, and it continues on to the distal convoluted tubule, which is the next step. And in that stage, essentially, sodium ions from the liquid are then reabsorbed back into the blood, and also potassium ions, hydrogen ions, and ammonium ions are moved into the tubule. And so essentially that's the important step of the loop of Henle. It's to get water out of the out of the loop of Henle, out of the tubules, back into the blood. And it does that by using sodium ions essentially to change the water potential gradient to get it to diffuse out. And so that's how water is controlled in the loop of Henle. So now we've looked at the loop of Henle, we then look at the distal convoluted tubule, and that's essentially just where more stuff is reabsorbed and put back into the distal convoluted tubule. As I said earlier, sodium ions are moved out, potassium ions, hydrogen ions, ammonium ions are moved in to then be excreted later. So I hope this video has been useful in terms of an overview of the kidney and how each stage kind of works, especially the glomerulus with the ultrafiltration process, the proximal convoluted tubule with the large surface area, the selective reabsorption of glucose, amino acids, important molecules that the body doesn't want to lose, and the loop of Henle, where a sodium ion concentration gradient is created to cause water to diffuse out back into the blood by osmosis. I hope that's been clear. Please like and subscribe. Stay tuned for more videos from Biology Interest. Thank you.